Bismillahirrahmanirrahim. Bismillahirrahmanirrahim.
Thank you, Dr. Safi. Good.
So Spectrum offers a range of UIs for setting up of search parameters as well as visualization of search results. It offers a customizable scoring scheme and offers support for unknown PTM. Spectrum offers support for single file search as well as in batch modes, and file formats that are supported by Spectrum are standardized. So to talk about the overview of Spectrum workflow, the Spectrum workflow, the pipeline begins with the input of MS spectra, which is in the form of raw data and processed into file formats which are supported by Spectrum. The first algorithm which the spectral data encounters is intact protein mass tuning. So in this algorithm, the spectral data is first used to form triple sums, which are fragment pair sums. These fragment pair sums are plotted, and a window scans these fragment pair sums, which are then used to identify the maximum peak count. This maximum peak count is used for tuning the MS1, which is the precursor ion mass. This tuned mass is then used to filter out the protein database, as a result of which we get the first candidate protein list. This candidate protein list is then subjected to PhD extraction algorithm, which is the sequence-based filtering approach. In this algorithm, we take the spectral data in the form of MS2s and intensities, and these ratios are then used to identify the differences between consecutive MS2 peaks. If the difference corresponds to amino acid, it is retained, and consecutive amino acids are stitched together to form PhD ladders. These PhD ladders are then utilized for filtering the candidate protein list, and the candidate protein list is updated and subjected to the next filtering approach, which is in silico spectral comparison. Within this algorithm, the theoretical spectra, which has been generated using the fragmentation methods defined by the user, is compared with the experimental spectra. And as a result of these, we get the matches. These matches are then used to formulate the in silico score, which is then used to filter out the candidate protein list. The updated candidate protein list is then ranked and scored using the composite scoring scheme, which assigns weight to the particular algorithms based on what user has specified, as a result of which the proteins are ranked and scored. Spectrum offers a range of UIs for visualization of search results. To clearly lay out the features of Spectrum, we have compared it with the tools, and as is visible within the table, Spectrum offers a range of features which are down every tools. And it has been benchmarked against TDP tools, including ProSci, PCP, Topic, and MS Pathfinder. So Spectrum has, as this Venn diagram explains the search results for Spectrum. Spectrum has identified about 91% to 177% more proteins in comparison to the contemporary tools which are available. Spectrum is available for download at the GitHub link. It is an open source, open architecture toolbox which is available for use within the Proteomics community. So next, we have developed an open source GPU accelerated web-based proteoform identification pipeline, which is Perceptron. So Perceptron is the next generation freely available GPU acceleration platform for TTP. It derives its search pipeline from Spectrum and offers three main algorithms as that happens with Spectrum. It offers high throughput performance due to execution of optimized code in parallel on GPU, and it is available at perceptron on lums.edu.pk. So this figure here outlines this general workflow of Perceptron. As happens in Spectrum, the spectral data is then first subjected to in silico intact protein mass tuning. Then it is subjected to PhD filtering and moving on to in silico fragmentation, as a result of which we get the updated candidate protein list. This is what the UI looks like. Perceptron offers intuitive graphical interface, web forms for input of search parameters, as well as for visualization of search results. Talking about individual algorithms which have been implemented in GPU. So the first algorithm which has been implemented in GPU in Perceptron is the intact protein mass tuning algorithm, which is the mass filtering algorithm. So in the first step, the spectral data is that is via a kernel. Kernel has a grid with blocks. Within each block, there are multiple threads which run in parallel and compute fragment pair sums. These fragment pair sums are then sent back to CPU for filtering based on the user's specified tolerances. As a result, in the third step, these filtered tuple sums are sent back to GPU for calculation of tuned mass. This tuned mass is then utilized for mass-based filtering within which is first transferred to CPU for onwards use. The next algorithm which has been developed in GPU is the PhD filtering algorithm, which is the sequence-based filtering algorithm. Within this, the spectral data is being transferred from CPU to GPU and within via kernel. This kernel again has blocks and threads. Multiple threads compute the differences between the consecutive amino acid peaks, spectral peaks, and if that mass difference corresponds to the mass of an amino acid, it is retained, and these PhD ladders are stitched together. So the using these PhD ladders, they're first transferred to CPU, where they're filtered based on the user's specified PhD length.
So what's this intensity?
Intensity is basically, this is spectral data. It has MS2s and the abundance of these MS2s, which is reported as intensity.
What is MSO? M over Z?
Ratio.
Charge.
Yeah, mass to charge ratio.
Yes, charge ratio. And then intensity of what? Is it vapor? What intensity is coming out?
Intensity of that spectral peak that has been observed within mass.
Based on mass charge.
Yes. So these PhDs are then used for onward filtering of the candidate protein list. So the runtime performance of perceptron was compared with spectrum, and as is visible in the table here, perceptron has outperformed spectrum by 10-fold in terms of runtime. For spectrum, the runtime performance for one data was 13,000 milliseconds, while that has been reduced in perceptron, and it is now 1,300 milliseconds. So now that we have developed spectrum and perceptron, which are the TTP analysis pipelines, we have developed another novel algorithm for identification of proteoforms within the experimental data, and for enhanced proteoform identification and characterization with top-down proteomics. So the successful spectral interpretation relies upon the identification of fragment ions. What are these fragment ions? Within mass spec, the proteins are fragmented, and as a result of each fragmentation event, two product ions are formed. So TDP has multiply charged precursor ions, which have been utilized through soft ionization methods, and the addition of these multiple charges increases the chance for each of the fragment to get detected. However, there are certain abnormalities which include neutron losses. So basically, one of the precursor, if the precursor ion is fragmented, one of the daughter ions gets all of the charges and the other one is not reported due to lack of charge. This is referred to as neutron loss. And ideally, all of the fragments should get detected. However, in reality, the fragments are not formed with uniform abundance.
And then what's the difference between the fragments in bottom up and the top up? So there also you have fragmentation, and there also you have.
In bottom-up proteomics, we have proteolytic digestion, we have peptides.
Yeah.
In top-down proteomics, there is no proteolytic digestion. We have fragments. So fragmentation is random, while in
This fragmentation takes place by how?
there are fragmentation methods, there are fragmentation techniques including collision ion association, there's ETD. So this fragmentation event is totally random. But in the case of bottom-up proteomics, we have specified residues at which the proteolytic digestion takes place. So every time we'll have certain type of peptides. But for fragments, we can have random fragmentation events, as a result of which we can have different types of fragment ions.
Yeah, so there's fragments and how these fragments are generated. After injecting into mass spec, they're exposed to what these fragments.
So there are different techniques, one of which is collision-induced dissociation, in which there's a chamber, there's inert gas, these ions can collide randomly, and after which some of these ions, when they get charged, they're moved out of the chamber and they get detected. So this is how the fragments go.
But there's also same kind of fragmentation in bottom-up. This is the basis of MS2.
Right.
So that fragmentation.
But that fragmentation, yes, sorry. That fragmentation is of peptides.
Doesn't matter. So the fragmentation phenomena is the same. The principle is the same. Right? So that's also I know.
The input is different. In this case, input is open. In that case, input is not. So the fragmentation in the top term is of the MS.
Correct. Intact.
Not of the protein itself.
MS is basically the protein.
Yeah, I mean, but you don't fragment the protein itself. You take the mass of the protein and you fragment that mass.
No. So that protein is fragmented. Computationally, we have the masses, but in actual, this computational data is coming from the spectral data in which the proteins have been fragmented.
First, the protein is detected as a whole, and
Yes.
then the protein is.
The two mass analyzer basically, MS and O.
Yeah, there's so that's what I wanted to learn. So in proteolytic image, we will have many fragments based on the, if you have used trypsin, they have many fragments based on the trypsin sites within the protein. Fragments, let's say, protein of 200 kilodaltons. And now you won't have the fragmentations based on the cleavage sites of trypsin.
Correct.
So is it random? Let's say you still have many fragments of the protein which is 200 kilodalton. Many small size because of this collision of the protein with the gas. Or you have bigger fragments, let's say 3 or 4 fragments of the protein.
We'll have many fragments.
Many small fragments.
Yeah.
Combination of R.
So every collision, so each peptide or a protein gets one cut or molecule, and that is random.
So there are not multiple cuts on it.
Not on the same molecule.
I'll show one diagram.
Have 2 fragments.
Yeah, so it's like a rubber band. So you stretch the rubber band, yeah, and if you cut it, so the strings they collapse. So you just get 2 portions out of a protein. 1% of the time, if it so happens, you might have a side chain form. So we ignore that.
Sure. And the questions here.
And maybe this diagram would help. So every time there's a fragmentation event, the entire protein has 2 fragments. But
That's
this
what
fragmentation
I
is
remember. I remember this time. That's why I asked you there.
So is there a size limit to the total mass of the protein? Because you have the first one where you take the total mass. So is there a size limit to that?
For high resolution mass spectrometers like FTICR, we can identify protein in several kilodaltons. But the range is in high resolution mass spec, it's automatically defined. We get a range of MS1s. We filter them out, separate them, fragment them, get the product type spectra, and then we define another range. This happens automatically.
But several kilodaltons is a very
The range is very small.
Yeah, like
In which we
100
fit it.
kilodalton protein, 200 kilodalton. What is the
That's the maximum size limit of a protein that can be analyzed,
Yes.
but the range is smaller. The range that is defined
range kahan se kahan? So range is usually from this to this.
depends on the type of a protein that is analyzed.
So let's say if I have a protein that is 100 kilodalton, so that will be analyzed or top-down proteins.
Of 500 kilodalton.
Yeah.
So you're talking about the, sorry, I could not understand.
I'm saying, with the top-down proteomics, you told us that you measure the size of the mass of the entire protein, and then you cleave them into random 2 fragments, and then you measure the mass of those, right? So what I'm saying is that the mass of the protein, is there a limit to that? Okay, कितने kilodalton इतने amino acids की जैसे protein उसका आप कर सकते हैं mass, इससे ज़्यादा हो तो अभी उससे नहीं करना।
Yes, there is a limit to that. It depends on the type of mass spectrometers which are used.
Yeah, so what is the limit?
I need to check it back. So, talking about the neutral losses, this is the precursor ion which has two charges, and one of the daughter ions which is formed retains all of the charge, and the other one is not reported due to lack of charge. This is referred to as neutral loss, and other possibilities could be missed fragmentation sites As a result of the neutral losses, the algorithms which are entirely dependent on the fragment time information, they suffer due to missing complementary ions. So,
Go back, please. I wanted to read the last answer. What last line means de novo sequencing and PTM identification suffers advantage of missing component. So we are at a disadvantage of identifying PTMs using
yes, if there are loss of ions. If there are loss of ions.
if there are loss of ions. So to identify PTMs, we don't recommend using top-down.
No. So top-down proteomics does characterize proteins. That is, they identify PTMs. But sometimes, if the spectral data resolution is low, these fragment ions are lost. So it entirely depends on the type of spectral data that is being analyzed. So for that purpose, we need more information to characterize PTMs. And for that purpose, we need to develop a strategy that helps us to incorporate those missing ions. So the loss of information due to missing ions is a challenging computational problem, specifically for algorithms which are entirely dependent on the spectral fragment ion data. So coins as from TTP data, which adds missing information to the spectrum. So incorporation of these unreported ions into the peak list makes it more denser and helps us to identify lower PTMs, enhance PSD count, improve PTM prism counts as well. This figure here on the top tells you about the experimental spectra which has been reported. And the figure at the bottom is the complementary ion spectra which has been generated through the implementation of Coins algorithm. So Coins adds missing complementary ions within the spectra and makes it more denser, which is now rich in information.
So go back. Sorry, I'm interrupting. So the bottom chart is basically is in the top.
Correct.
And you have reported missing ions, which you are saying is one of the limiting factors for PTM identification.
Correct.
How you capture those missing ions from this data?
So if we look here, the precursor ion, every fragmentation event would lead to 2 fragment ions. And the precursor ion, when it is fragmented, if one of the fragment ion is reported, the remaining is not reported. We have formulas that calculate the sum of these. So if we subtract this complementary ion from the precursor ion data, we get the remaining data, fragment ion which was not reported. So this is the basic strategy that was implemented. If we have one of the fragments reported, we have precursor ion mass, we subtract this fragment ion information from the precursor ion mass and get the information that was missing.
Sorry. So let's say precursor ion MS1 is available to you.
Yes.
The other one is missing.
This is also available.
That's also available.
Yes.
Okay. If both are available.
Yes.
So what was missing then?
Second fragment ion.
Second fragment ion. Lost.
We don't have drop. The MS1, which is the intact protein mass. We have one fragmentation. We have this reported, which is the intact protein mass, which is then separated and fragmented to get the product ion spectra. We have this daughter ion, which is one MS2. Let's call it MS21, and let's call it MS22. So we have this ion reported, we have this reported, right? And this was missing. So to calculate this, we subtract this information from this, and we get this.
So basically, you have MS1 and MS2.
Yes, one of the MS2 is reported.
Thank you very much.
So, Kanzal, I think it would be useful to elaborate why would such an event occur? Why should we have an uncharged species coming out?
Yeah. So for that purpose, for instance, we have multiply charged intact protein. This charge is retained by one of the fragment ions, and the other one was not detected. Why? Because mass spectrometer only detects the charged species. So if there is no charge, this would not get detected.
But when you subtract that, you get this neutral fragment, but then you validate that in the data that yes, it is there.
Yes. So one
But,
strategy for
but
that.
you said it's not charged, so MS does not detect this.
Yes.
How do we detect this neutral one after using your method? You know, taking the full length minus MS1 data minus MS2, you get the MS2 too, okay? And this goes missing. How you validate after your method that this is actually present in the.
So one strategy for this is basically we exhaustively calculate MS2s using the experimental spectra that we have. So it is the fact that we have some of the MS2s missing, but most of them would be reported. So if we get the MS2 which was already reported, we do not add this information back to the spectra. So this is one strategy we validate that this calculation is right. Also, this algorithm has already been implemented in the bottom-up proteomics. So in top-down proteomics, it was not implemented.
So that we call it pseudo spectra there. We don't call it MS2 spectra or MS2 pseudo spectra because it's not real from the experiment.
So, some parts of the protein or some fragments, microterrains, right? Next charge will be. So they won't contribute. I mean, correct me if I'm wrong. They won't contribute any charge to MS1.
I don't know. Would you please repeat it?
So maybe part of the fragment in the MS2, that part is a current charge.
No, so it will have
Or is it only
overall
that it
charge.
doesn't detect the charge?
It will be overall charge. It will.
Overall charge, one part of the fragment in MS2. If it doesn't have an overall charge, it's small, few millions, it doesn't have a net charge, right? So that part is not contributing to the charge of the whole protein in the MS1 as well.
Correct.
Right? So what will be the result if you subtract that to the penny market? You know what I mean?
So in order to utilize the information within the spectral data, we decharge it first. That is called deconvolution. We first convert the spectral data into charge 1 state and then use it further. So multiple charges would be deconvolved into charge 1 state. Does it answer your question? So Points has supplemented the proteoform search process by providing information on the missing ions. These graphs here represent the proteoform count at 1% FDR, prisms count, and PhDs count. So there was an 86% improvement in the proteoform identification using points approach. These bars here depict the endpoints, overall endpoints from 2 different types of spectra, ETD and CID spectra, and this is the points approach. So points has improved the proteoform search by 86% improvement. The prisms counts, which are the proteoform spectral matches, were enhanced by 98%. and the overall PhD count was increased by up to 2-fold. So during MS, fragment losses are dependent on the fragmentation method which is employed. So PTMs for spectral data set that was generated using alternate fragmentation methods, which incorporates ETD type of spectra, CID and HCD. So Points has reported novel PTMs and enhanced the PTM count by up to points, endpoints. Endpoints is basically no points approach.
Which data you use to do all this?
Published data.
Published. And this published data already tried to take some PTMs?
Yes.
And you identified some more PTMs?
Yes.
These were PTMs just, that this group actually identified that on this particular protein, this PTM exists.
Yes. So this algorithm was implemented in Spectrum, and Spectrum does offer support for identification of PTMs within. It does report the site at which the PTM takes place, as well as what kind of a PTM is there.
Yeah. And using coins, that identification you asked.
Yes.
And was it mammalian data? Which data?
So this was the bovine data set. Whenever we get, for example, the proteoforms, we got it at a stringent or proteoforms. This proteoforms at 1%.
Using this data, using coins over your approach, could you filter some known PTMs which are reported, which are known functionally relevant? That will have significance to the computation.
Correct. So that TDP is basically there are very limited set of data sets which are publicly available. So when we talk about published data sets, it is very challenging to come across the data that has the parameters made publicly available. So if we get the spectral data set that incorporates the parameters that we are talking about, this will help us to implement the points approach.
So I'm saying P53 protein. In this data which you analyze, can you fish out this is P53?
Yes.
So you fished out P53. And P53 has many reported post-translational modifications or any XYZ protein. And you can see how many of the reported ones are actually being predicted or computed using your approach, and are there some novel ones which you predict?
Right.
Did you get my point now?
Yes.
What I'm asking?
Yes.
How difficult it is to do?
Not really with the points approach.
Yeah.
I think what Tariq is trying to say, for example, you want to see phosphorylation sites in a protein in the cell. So what you do is you isolate that protein from the cell, do a proteomics analysis, you identify there are 10 phosphorylation sites, right? And then you mutate those sites one by one and do a kinase assay, and you find that actually only 2 of them are phosphorylated, not all 10 of them. Right? So this is validation. So what it is trying to say is, let's say you identified 1.5 times more phosphorylation sites. So is there a way to validate that? I think one way could be looking to the literature, proteins that were initially mass spec to identify new phosphorylation sites. How many of them were validated? So, or the ones that were not known and the mass spec identified. Both ways. You can try to find those, or somebody after you completed a lot of work, but somebody could validate.
Yeah. Thank you. Thank you. That's a very good suggestion.
So just another question around. So you said in the beginning the spectrum cannot do quantification.
Yes.
But I mean, there is a level 3 quantification which is very popular. Why not implement it in the, you're also context, you're doing spectral counts.
Yes.
Why can't you implement the, I'm just wondering, is it too difficult or it's
No, it's not that difficult. Basically, we've not started off with the quantitation algorithm.
You can modify that. So
We'll look into this. Alright, so points is available for download at GitHub. Talking about the second research objective, which was structural proteomics. Now that we have developed an open source software and tool for the sequence-based analysis of proteoforms, which alone is not sufficient to identify the function of proteins. So we'll have to look into the structure and devise a strategy that helps us to understand the relationship between protein sequence and structure in order to find out their functions within the organism. So we have developed an application of structural proteomics, which is basically an integrative in silico framework for sequence structure-based proteomics. Application we have chosen for infection that element. So NS34 is basically a very difficult therapeutic target. It is a multifunctional protein, and due to genotypic diversity and resistant mutations, NS34A impacts the efficiency of NS34A inhibitors. Specifically, there are mutations within the ligand binding pocket of NS34A genotype 3, which is prevalent in Pakistan, which causes resistance in the drug binding, leading to the emergence of drug-resistant variants. So, frequently observed RASes have made genotype 3 naturally resistant. Moreover, there is lack of availability of genotype 3-based NS34A crystal structure, which further limits the investigation of the specific mutations and their impact on the inhibitors. So, this figure here represents the NS34A crystal structure. So there are 3 catalytic triad residues, and there are several binding site residues. So the protease inhibitors, they attack serine 139. As a result of this, they block serine 139. The catalytic site is blocked, and NS34A is stopped from working. So mutations within the ligand binding pocket imparts resistance to these inhibitors. So for that purpose, they have developed a structure and functional analysis of this protein. To begin with, we have retrieved sequences for HCNS34A and identified the binding site. These sequences were aligned to identify the sequence variations. And on the other hand, we have identified the template for modeling the structure of these sequences. So a homology modeling approach was used and the models were refined. So this figure here represents the mutations within the ligand binding residues of HCNS34A. As can be seen here, 2 mutations were identified within the catalytic triad, and 14 mutations were overall analyzed. These 14 mutations were incorporated into one each variant, and as a result, we have 14 variants, which were then used for further analysis. The models generated were structurally assessed, and the analysis tells us that the residues are within the favored region. In the second half, we have worked on the integrated workflow, which is the pharmacophore-based virtuous cleaning. And in this process, we have isolated the features of drug-like compounds, of approved drugs, And these features were then used to develop a pharmacophore. That pharmacophore was used to filter out the databases of compounds. As a result, we got several thousand conformers, which are then filtered using different filters, including Lipinski's rule of 5. And depending upon this filtering, we got thousands of conformers, thousands of compounds. These compounds were then prepared to dock with the protein variants that were modeled. So the ligands were prepared and the protein was prepared for docking studies. The docking protocol that we analyzed was covalent docking protocol. As a result, we got 23 compounds which were making interactions with the protein variants that were modeled. So these compounds were then subjected to post-docking analysis, in which we identified the ligand-protein interactions using Maestro Schrödinger, and the conformation analysis led to the identification of 7 compounds from these millions and thousands of compounds. So these 7 compounds indicated good interactions with the protein variants that incorporated mutations. And then these compounds were subjected to molecular dynamic simulations. And as a result, we identified only one compound that indicated good binding with the mutations which are drug-resistant mutations. So this figure here represents the pharmacophoric features. As a result, this compound was isolated with so this figure here is the molecular surface interaction diagram. This tells us about the ligand interacting within the ligand binding pocket of NS34A. And these diagrams here tell us about the ligand interaction within the binding pocket. These are the residues, and this indicates that the ligand is basically covalently bonding with the residues the binding pocket of the genotype. The calculate MDBSK, which is the energetic interaction analysis, comparing the wild type energy with the variants. And lastly, we analyze the residue. And these bars here tell us about the interaction of these variants with the compound that we have isolated.
So did you identify 18 variants?
14. 14 mutations and 14 variants.
So this library that you've seen, how big was that? 100?
Which library actually?
So did you screen a library of compound or just you made a pharmacophores?
We made pharmacophores to screen the compound libraries, zinc, PubChem, and the available compound libraries.
Okay, so how many compounds were there?
There were several thousand compounds, and the pharmaco features that we extracted from FDA-approved drugs, they were used to filter out those compounds. And as a result, we got several 100,000 conformers, which were then subjected to a series of
filters. You got a final one compound?
Yes.
So is it commercially available?
No.
How is it covalently binding?
Sure. So the docking, no, it's covalent docking. It's a recent protocol for identifying inhibitors that bind covalently with the proteins within the binding pocket. So there are 3 catalytic triad residues with an ACVNS3, and there are 7 ligand binding residues. So they're interacting with those ligand binding residues covalently.
Some cases are covalent.
Yes.
How is it?
No, he's
No,
saying it's a new methodology, which is basically that's common. Yeah,
So for every variant, it is making one covalent bond with the residue.
Okay.
But all variants are making covalents.
Okay. So which residue is the bonding there?
Um,
Is this compound commercially available?
no, it is available in Campbell.
Can we get it synthesized? Inshallah.
Yeah. One, there is another patent to screen has this acid. And one was, I will use it on synthesize the compound, finish the test.
Right. Right.
Outside. I think this is interesting to show what is the chemistry.
So is it the same part of the molecule that binds to different residues? The part of the compound that makes the covalent bond is the
Yes.
same part of the
Yes, mostly these inhibitors, they make the covalent bond with CD139, which is a catalytic triotricity. 139.
So it is a
CV.
Yes.
Can you go to the protein structure and see go
Yes. Yes.
Yeah.
So like this CV.
Yes.
So how do you think that CV, which atom of the CV will possible binding which particular atom of the
So serine has the hydroxyl group, which attacks the warheads of the compounds, which are, sorry?
what is the one?
There's an electrophilic attack.
compound पे जाएगा उसको बताए कि सीक्वेंस इसके साथ which part of the
Yes.
So now CV, you have this hydroxyl group, where is it acting?
Can I open another presentation? I have an elaborate presentation.
Yes.
For that.
yes
वो तो 11 मैसेज है ना ठीक
How do I disconnect this? I need to disconnect this so that I can open another PowerPoint. Yeah, I correct. But I didn't connect it. Yeah, I need to find out the PowerPoint presentation. Of proteins within these microbial species. So you can, this research work proposes a multi-level proteomics pipeline that brings together different computational approaches towards sequence structure and function analysis of proteins. First, we have developed Spectrum, which is an open source, open architecture meta toolbox for TTP analysis, and it has enhanced the proteoform identification in comparison to the contemporary available tools by 91 to 177%. This is a novel composite scoring scheme which has been designed for 3 leading proteomics algorithms, and the scoring scheme helps to tune the process based on the filtering weighing of each of these component scores. This has helped Spectrum to perform better in comparison to the existing tools. Next, we have developed a based platform, which is an open source, deeply accelerated TDP search engine. It has outperformed even Spectrum by 10-fold in terms of runtime and identified up to 135% more proteins in comparison to the available tools. After this, we have developed Points, which is a novel algorithm for improved TDP data analysis. Points has enhanced proteoform search by reporting 86% more proteoforms, 98% presence, 3.6-fold increase in the PhD count, and significant increase in the PTM characterization along with the identification of novel proteins. Lastly, we have developed an integrated in silico framework for protein sequence structure and function analysis, for which we have used HCVNS34A as a case study. GC-specific drug-resistant mutations at 14 different positions were identified, which were modeled for in silico analysis. The potential covalent inhibitor of drug-resistant genotype-specific HCV-MS348 was identified, which represented stable binding with the residues within the ligand binding pocket. The translation proteomics case study identified microbiota residing within the wastewater channel, that is Hadiara drain in Lahore, Pakistan. So conclusion is basically that the proposed pipeline paves way for developing a next generation integrated in silico platform for protein analysis. The limitation for this this overall work is basically that the entire work, spectral data work, is dependent on the spectral data, input data, and the quality of spectral data basically determines the efficiency of these algorithmic pipelines. The type of fragmentation method employed during MSMS, which is the tandem mass spectrometry, and the resolution of the mass spectrometer. Moreover, the integrated in silico framework requires wet lab validation, and an extension of the proteomic case study can identify proteoforms that impart resistance to the microbiota within the environmental stress. These are the publications of the chapters that I've explained, that I've contributed in. The 3 papers are in review, while the others have been published. My contribution to these case studies and pipelines. Lastly, in the future, we are planning to develop the GPU implementation of these algorithms and design of algorithms towards handling complex modifications and enhance PTM identification molecule through database of modifications. Lastly, I would like to thank my supervisor, Dr. Safila Chaudhary, for immense support in my final defense committee. Dr. Shekhar Mirza, Dr. Muhammad Tariq, Dr. Shimshad Zamin, and Dr. Basil Shafiq for their valuable feedback and comments. Sir Sayyid Babarali for SPR Research Report 2021 and Dean Dr. Sabi Anwar. I'd like to thank international reviewers for their feedback and comments that helped me improve my thesis work, and the Faculty of Life Sciences Department for being a huge inspiration, our international collaborators, Team Proteomics, Team HCV, Royal members, and the staff here, LUMS and HEC for providing me with an opportunity for this PhD program. Thank you.
So questions. The house is open for all kinds of questions, basic science or regarding Kanzal's research, everything is open. So please speak up, especially the youngsters from birth. Okay, over to you.
I am the youngest.
Please. Okay.
So, that's a very nice presentation. Very nice way.
Thank you, sir.
Congratulations to you for the second place. From my knowledge, this some of the rest, when you mentioned this identification of post-process vision and what is algorithm and data, the most common PTM is cross-combination. And many times it happens that aspect which is there, it is dropped out during this process. And the P and P is phosphorylated part is PA value is not phosphorylated. But the people generally must to overcome this problem. If they are specifically looking at phosphorylation, they do a bunch of chemical modification, which helps to avoid this problem. So, did you incorporate that aspect?
No, we didn't.
Because otherwise, especially this kind of, because other post-translation modification are quite stable.
Right.
The phosphorylation has this mentioned problem that we cannot be precisely detected. It always gives misleading.
Right. So within spectrum, what we do basically in all the algorithms, what we do is we calculate the mass shifts.
Right.
If that mass corresponds to the modification, we then check for blindly check for the localization of that.
Right. But there is possibility that the particular maybe CD was phosphorylated, but we don't rush because the phosphate was not. That's what I
No, I wasn't aware of that. I looked into it.
So related to this question, this program will only identify the known, videos. Does it have any kind of prediction power that it can predict if you have samples from very different treatments or states?
Yes.
And in some cases, the protein is totally unmodified, and in other case, partially modified. Last case, we are fully modified. So I'm trying to understand whether it has any potential to kind of tell you that there was this modification, but it's not present in the unmodified one. So to say novel identification or prediction.
Yes, novel in the sense which are already reported, PTMs which are already reported.
No, reported are the ones which are known. Novel which are still unknown. So there are many things which are still unknown.
Yes, so the blind PTM module of spectrum identifies unknown mass shifts. It tells you about where the mass has been added. It tells you about the localization of that PTM, but the mass shift is identified blindly within the PTM.
So just to, this is just a tangent. It's an excellent area that we're planning to explore. But for now, we just report the mass shifts. So we just report the mass shifts. We cannot, at this moment, link those mass shifts to a molecule. But yes, in the future, we're planning to do some kind of, AI on that. So if you can map it, that makes sense.
maybe non-specific molecules, which are not just chemical molecules, for example, that glycation.
Yeah, actually, Kanzal missed one point, so let me add it. So we do have chemical molecules, NMEs on the anthem, oxygens. So we have certain chemical molecules. It's not a very large repertoire of them, but we have 4 or 5 chemical molecules. Therefore, of course, we can expand that list by adding more. It's just a mass shift for us and a modification for you.
For these algorithms, no, it is specifically designed for data analysis of mass spectral data.
So, neutral those that are defined, you just identified that those fragments who are made, which fragment is a part of which protein is on.
Yes.
You just identified that these are the those fragments in a data set. So we can mention that this those fragments in data.
Yes, so the precursor ion basically corresponds to the mass of that protein. So we isolate those precursor ions and then get the fragments out of them. So we already know that this fragment is coming out of that precursor.
What is the voltage of covalent inhibitor? Number one.
Very strong bind.
And number 2 is how would one experimentally determine the affinity of this compound covalent?
So to answer the first question, it is a very strong binding. It can be reversible or irreversible depending on the type of the compound. Secondly,
Is it only for the strong binding?
to inhibit ACVNS3 yes, to block it. And to answer the second question, experimental validation, I'm not aware of that protocol. I'll have to look into this.
Post-translational validation, but when you come across all cell type for medical specificity, when
you take that into account?
Yes, so PTMs are different. They help the localization of proteins within different parts of the cell. So they impart function to those proteins. So they differ depending on their localization within the cell. And
Talking about cell types.
sorry?
Talking about cell types. Your layer cells,
Yes.
your skin cells. Will you have similar kind of process? The protein, an X protein can be in this cell. It's not very different on skin.
That will be different.
So how to take that problem in this kind of analysis?
So the spectral data set that we currently, the spectral data set that we use is already published and we know what kind of protein is there. So does that
What
answer
you
your
would do to extend it is that analysis.
I would have to check that and think about it.
More questions from public?
Yes.
So, अगर से जो
For the algorithms you're talking about.
yes, I think for last one is
Sure. Yes. 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 So these. So the drawback with the first toolbox was that it was MATLAB based and it required a MATLAB license and it was time consuming. So to overcome that, we have developed Perceptron, which is web based and requires less time. So the runtime performance was improved. And to improve these two toolboxes search engines further, we have developed Points, which enhances the proteoform search even further. So this is what we do.
Okay, this is a very nice presentation. Thank you. Very well done, project. Just have a comment. Wondering if you mentioned one person for the other. And then you have like your data is distributed from another which have predicted these PTMs. And then you have had your data.
Yes.
As well as I understand, data is post-processed. So then the validated, so something predicted something.
Yes.
Validated and predicted these. Don't you think it would be better if you have some experimental validation?
So
And then also select and then why would you select one person? I understand what it means.
Yes.
But why then just 1%?
So the first one, what we do is we take the data sets, we benchmark them against the available toolboxes, but we consider this that we replicate those search processes and identify the proteins reported by those toolboxes, keeping in mind that both the parameters for our pipeline as well as the pipeline engaged are similar. Second, the experimental validation for this overall process. So we are talking about the computational pipeline, so we have not looked into the experimental validation for this.
Okay, so you're just trying to answer yes, whether you predicting whether it's more activated than anything you can't not that you wanted to do that in the case, you have somebody else in the lab maybe.
FDRs, false discovery rates, they are post hocs, and the best they can do is to provide us with an arbitrary resolution of error. And 5% is accepted across the world for proteomics at least, and I know for other fields, AMR as well. But we wanted it to be 1% FDR because it's a new platform. It's not an established platform. People have their questions, reviewers, they, trash us all the time. So if we are equally stringent, so this would probably not give them an additional piece of confidence, a bit of confidence to kind of use spectrum for this. But if we have a higher stringency on our tools, so this will give them, so FDR is blind, right? So it wouldn't care if it's a correct identification or not. As soon as the two distributions overlap, it's going to put a cut. So this makes our tool a little bit, more reliable. Like you say. Yeah, so they have it at 5%. So we are.
Can you use this pipeline for bottom-up proteomics data? Because the principle behind it is the same. I mean, you're looking at one fragment, it could be a large peptide, it could be a protein.
Yes.
And then you're looking at small fragments. It's the same principle.
Principle.
In principle, it's the same.
Yes.
So, can your pipeline be used for
It can, because essentially the principle would stay the same, but it would make the data more complex. Because dealing with proteins has its own complications, but dealing with peptides, they're very hard to recover. I don't think so personally that it will identify the same, the efficiency of algorithms would stay the same, depending on the data that we have for
maybe that's a litmus test for your pipeline, because that's a very noisy data. And if you can, confidently identify proteins and PDMs from that data. But I mean, it's very frequent that you have missed cleavages. Peptide digestion or lysine, including and lysine are used for. Then we get missed cleavages, which makes some large peptides. Maybe you can, looking at them as one data and they the large peptides, consider them as proteins, and then run your pipeline and see. Well, I mean, there are pipelines available for bottom-up data analysis. Some of them are free as well, but I mean, if your algorithm works better than the other contemporary outcome, maybe bottom-up as well.
You can move into observation. Thank you.
So why do we use FDR?
Sorry?
Why do we use FDR?
To validate that the data that we have identified is correct.
And how would that happen?
So FDR calculation basically in a generalized statement, FDR calculates the false positives. So we allow for 1% FDR. That means in every 100 isolations of every 100 results, we allow for one result to be false positive. So that's very stringent. Does that answer the question?
I think to offer a better explanation. So the two distributions that we have, decoy and
do I make it here?
Am I allowed to do the cheat? So yeah, you can draw. So it will help them understand.
All right.
So maybe they can apply these tools to their.
This is going to go
Yeah.
Decoy basically in this case is the shuffled database that incorporates for spectrum. It also incorporates 3 types of mutations. So it's a very random DB. Chances of this random DB providing us with the proteoform identification should be very, very low. So this is the 1% FDR threshold. We allow for one false positive. This is the target DB, which is the protein database. So what we do is for every search, we set up this threshold to be 1% FDR. we allow for one false positive identification, that is of the proteins that we have identified, for one protein identification to be wrong. That is what we do for spectrum and reciprocity.
So does it in some way answer to what Dr. Shoaib was asking, that you use bottom-up data? It would sound more random.
Yeah, so
So technically we use which is kind of, so it can't be worse than that. Yes, so bottom-up would still have some aspects that are biologically correct. But before is a totally random. So that's why I think we have a better control.
I understand that there would be a very clean difference between top-down data and the decoy, and the difference is because the bottom-up data is also close to random. So then your FPR 1% probably too strict.
Yeah,
Maybe 5 or 10%.
yeah, absolutely. Maybe we could identify more PDs at 5%, but just for the sake of the manuscript,
So what is next in your HCV project? What do you propose? Where should the lab focus on?
So one way could be the witness validation for the conform that we've devised. If that compound is validated, that would help us identify. So we've already reported the drug resistant mutation. So for gene type 3, which is prevalent in Pakistan, that would be a huge contribution towards.
You said it only binds to a specific seeding variant.
No, it binds to all of the variants that we have.
It binds to all the variants. If
Yes.
this thing is conserved, all the variants in?
No, so one. I'm showing you. Yes. So we did mutation. Serine 139 to proline.
So this seed in 139, so your compound binds there.
It for this variant, I'll have to check with 14 variant. It binds to no. So what happened was, for this variant, we could not do covalent docking because we were essentially looking for the covalent inhibitor that binds to serine 139. For this purpose, we did molecular docking.
These 14 variants came up from how many total samples or how many?
200.
And how frequent this site gets mutated? How frequently this seeding where this particular compound binds?
There was only one variant we could identify, which is represent. Basically, these are the representative tests for the collection of mutations. So we did identify this mutation, but I'll have to check back how frequently this was mutated.
And when you look at all 200, how frequently this seeding is conserved or?
It should be highly conserved, but I'll have to check it.
Because out of these 14, you have only one control spot. All the rest are CXs. So can you go to the very early stage experience that proteomics is? Oh yeah. Saying what this, you have the grids, blocks, and then you are having certain scores, which I assume are these intensities. Whatever. So what is a single block? You are zooming in, and you are showing us these. Explain this. I could not learn this.
Right. So one has these multiple blocks. These blocks contain multiple threads which can run in parallel and compute different values. So we utilize these threads to compute fragment pair sums. Every thread will compute the fragment pair sum. And essentially these fragment pair sums would be sent back for filtering in the CPU. So all of the computation is being done in GPU. So it requires, so GPU is is the high throughput, has high throughput performance. So all the computation is being done here, and for CPU we are only utilizing it for filtering process, which does not require much time. So in order to save time and enhance the runtime performance, we utilize GPU threads for parallel processing of the data. In Spectrum, what was happening was that all of this computation was taking place in CPU. And if if one thread was completed, only after that the second thread would be used. So that required more time.
And this is MS2 data?
Yes, yes.
This is these, you were telling us 2 fragments are
Yes.
being generated.
Yeah. So, Dr. Shamshad, are you with us? Assalamu alaikum, Dr. Shamshad. Is she with us?
Yes, Assalamualaikum.
वालेकुम अस्सलाम जी डॉक्टर शमशाद, उम्मीद है हमने आपको बोर नहीं किया होगा.
नहीं, आपने बोर नहीं किया. I truly enjoyed her presentation and most of the questions I had in mind she already answered. और बहुत अच्छा interesting काम है. It's very one thing I would like to ask her, by the way. She was referring to de novo sequencing as well, right?
Yep.
So I was like a little bit, I had my reservations because when you use the words de novo sequencing using proteomics platforms, there are like lots of other issues and concerns. Like, for example, there are a couple of amino acids which would show you exactly same mass and hence confusing the overall output. Similarly, like dipeptide, a gly is going to give you mass of asparagine. And that's why, like, being a protein chemist, this is always my concern, that how using MS fragmentation analysis, one can identify the exact sequence of a protein molecule. So I was wondering whether, do you have any additional identification platform or any additional criteria to judge that? Because I haven't seen any proper software to do that so far.
Right. So, ma'am, what we do in Spectrum and Perceptron in the search pipeline is for de novo sequencing, we incorporate, like you were talking about, 2 residues having the same mass, we incorporate both of these residues and derive 2 PhDs. And both of these PhDs are incorporated into the search process, and therefore we incorporate the isoforms as well. So to answer, I'm sorry, what was the second question about?
How do you detect such a case? जहाँ पे using isolation, so then
So it depends on the fragmentation that is being a method that is being employed. So there are several fragmentation methods like CID, ETD, ECD. So ETD is known for isolating the differences between such amino acids like isoleucine and leucine and valine. So it depends entirely on the type of spectral data that we have. So if we have ETD, we'll be able to differentiate between these types of mass residues.
Okay, that's interesting. That's interesting. Okay, you have been talking about validation of your data a lot. Obviously, this is one of the most important criteria whenever we are interested in implementing any particular software. When within the lab we perform proteomics analysis, we usually use any particular software, like which are establishment, of course, but we use various kind of like software which in which we have to specify the type of instruments we are using, the type of fixed mutations we expect, and type of mutations we kind of like expect happen, which might happen due to experimental results. In your case, in your analysis, I believe you still hit your data by collaborating with someone who is performing top-down proteomics and analyzing your data in terms of whatever they come across and then what your data is going to interpret. Is that so? Am I correct in assuming?
It could be one direction. But what currently we do is that we benchmark we have benchmarked our TDP tool according to the contemporary tools available. Also, to validate what we have identified, we basically run the search processes in parallel in order to identify that what we have given as an output is correct and has been identified by the other contemporary tool as well. So that is what benchmarking is about. That's how we validate the data set as well as the software that we've developed.
So, but in your limitations, you said you still need to validate it using experimental data, right?
Yeah. That I was talking about the integrated in silico pipeline that incorporated HCV-NS3 study. It needs to be validated that the compound that we have identified is experimentally showing the same efficacy.
Oh, okay. I see. I got it now. Okay. Thank you. Thank you so much, Kanzal, and all the best for your endeavors.
Thank you so much.
So, let the people sit here, we go out. Can we move the computer so that we have a...
I don't think take it. We should take it along.
So that we have a conversation and then we come back to the 5 minutes. So, pass it? Keep. So, yeah, please see here.
Kanzal, but have a seat. Shamshad, you're here?
Yes,
Hello?
I'm here. Very much here.
Okay, so we're going to convene with the committee, so please stay online.
Okay.
Congratulations. May God make your way easy and may you become a symbol of truth, honesty, and bravery that we so need. Thank you so much. So before we conclude, I'd like to request Kanzal's mother and Usman, her husband, to please join us here and have a group photo with Kanzal, and then we can discuss. So big hand for Kanzal.
I, Kanzal Iman,
solemnly declare
solemnly declare
that
that
I will always use my knowledge
I will always use my knowledge
and expertise
and expertise
for the benefit of humanity.
for the benefit of humanity.
I will never directly
I will never directly
or indirectly
or indirectly
get involved
get involved
in any activity
in any activity
that may harm humanity.
that may harm humanity.